Good afternoon. Today I will present this paper, Bioremediation on a chip, a portable microfluidic device for efficient screening of bacterial biofilm with polycyclic aromatic hydrocarbon removal capacity. In this paper, they determine the removal of polycyclic aromatic hydrocarbons with the bacteria Pseudomonas, P26 and Gordonia H19 as pure cultures and co-cultures. In materials and methods, they use the microfluidic design, the microchip design in both 4 channels with 4 sisters each, 1 inlet and 1 outlet. The microchannel design has 32.22 microliters as total and internal volume. It is fabricated with polydimethylsiloxane, PDMS. The first assay they realized it is the biofilm formation assay. Here we can observe a schematic representation of how it was the assay. First bacterial suspension were seed and recirculate in a flow rate of 70 microliters per minute for 72 hours. The biofilm structure was performed using electronic scan microscopy. A microscopic study was carried out after biofilm formation. In this assay, they compare with a slide of glass slides with identical material to the floor of microfluidic device in the same condition, like a control condition. Then the next assay they realized is how the hydrocarbon removal in the microfluidic device. Here we can observe in the same schematic. Once the biofilms have been formed, they recirculate the hydrocarbons with the culture media, and they analyze and they recirculate for 96 hours, 4 days, and then they analyze determined by HPLC. Okay. Here we can observe in this photography how it is the formation of biofilm inside the microfluidic device and in the glass slide that it was used for compare. Clearly, we can observe that in the case of Pseudomonas, Gordonia, and the mixed culture, the biofilm format is better than glass slides. Here in this graphic, we can observe the same too. The biofilm cover area is better in a mixed culture, but in both cases, with Pseudomonas and Gordonia, the biofilm cover area is better in a microfluidic device compared with a glass slide. Okay, in another graphic, in this graphic, we can observe the removal of the hydrocarbons, and in all the cases, the removal in a microfluidic device is better using a Pseudomonas P26. The removal was better of pyrene, fluoranthene, and acenaphthene. In another case, we can observe that in another condition that it was using planktonic cultures in a flask, the removal was better compared with a microfluidic device in the case of Gordonia, and mixed culture. Finally, we can observe that Pseudomonas was efficient removal of the hydrocarbon. In conclusion, they obtained a robust and pure and mixed biofilms cultivated and microfluidic platforms. They observed that Pseudomonas is so efficient to remove hydrocarbon because in Pseudomonas P26 biofilm, the one that showed the highest removal within 4 days. The next paper I choose is this, named the effect of shear stress on the formation of bacterial biofilm in a microfluidic channel. Bacteria has a extracellular polymeric substrate, EPS, denominated EPS, that protecting bacteria under physical and chemical conditions. And shear stress is an important factor to build a stable biofilm. For this reason, it is important to study the effect of this in the microfluidic device, to simulate the water flow in the same way that in a real environment. In this paper, they study the biofilm formation in a PDMS-based microfluidic channel, which is able to simulate fluid pips at a smaller scale. They study the bacteria Pseudomonas aeruginosa because it's one of the major opportunist pathogens and a model microorganism to study biofilm. Recently, biofilm experiments using microfluidic technology is successfully implemented. In this micro device, we can simulate the biofilm formation and grow under various shear stress. The microfluidic device for this study was fabricated using soft lithography technique, and it was a mixture of PDMS, prepolymer, and curing agents. The biofilm formation in a microfluidic device. Measure biofilm with homogeneous cells coexist has a strong resistance, although many research don't understanding the mechanism of biofilm development. For this reason, microfluidic systems integrate with living cells is a promising tool, because it can control the spatial and temporal condition of cell growth and environmental stimuli in vitro. In this essay, they prove the microfluidic in various shear stress conditions, and they determine the biofilm strongly depends on flow rate in microfluidic device. Here they observe how is the formation of the biofilm using optical fluorescence. We can observe that in the 4 hours, bacteria initial adhered and grew. After 12 hours, bacterial cluster was formed. And finally, a mature biofilm structure is developed and full cover in a microfluidic channel for 24 hours. Here in this figure, we can observe in a flow rate over 0.1 microliters per minute, the growing is better compared with a low rate. It is because initially with a high flow rate make that the planktonic bacteria have been removed, and it prevents a stable formation of biofilm. Clearly, we can observe in this figure. Here, the formation of the biofilm is better in this flow rate than in the anodes. Then in this graphic, they analyze how it is the percentage of cover area compared with the time of growing of bacteria. And in this graphic, we can observe the stable formation of biofilm, and it is developed at 0.1 microliters per minute flow rate. The coverage of bacteria biofilm in the microchannel is dramatically reduced in a flow rate such as 0.04 and 0.05 microliters per minute. We can observe here that in this low flow rate, the growing of biofilm is lower compared with the cover area. The cover area of biofilm at 0.2 microliters per minute approximately is about 80% of the entire area, like we can observe in this graphic. In conclusion, this study shows the significant difference in change of their morphology, and the dynamics of biofilm formation. It strongly depends on a flow rate in a microfluidic device. They determine in then experiments what is the value of the shear stress that make an optimum condition to form a stable biofilm. And for the observation of the image analysis, they use the green fluorescence image of biofilm. And for the surface cover of biofilm, it was calculated using Image Pro software. This is everything I presented. Thank you so much.